I'm Rachel Henning, and I had the pleasure of being the Becker Fellow in Research at the University of Chicago in Dr. Richard Craig's lab this past summer. Dr. Craig's lab is a neurology lab at the University of Chicago Medical Center that focuses on migraine treatment as well as treatments for multiple sclerosis. Here's the group that I worked with within the lab. Here is Aya Pusik. She is currently working towards her PhD. Here is Dr. Craig. Here is Lisa Wan, who was a lab tech in the lab. Here is Kai Pusik, who is a postdoc in the lab. Here's me, and here's Yelena Grinberg, who recently in October received her PhD. This summer I also got to partake in the Pritzker School of Medicine Experience and Research, also known as PSOMER. William McDade, shown here with me, is the founder of the PSOMER program, which provides students with research experience as well as the ability to shadow others in clinical settings. Here at the end of the summer we gave presentations of our research findings. These are my 3 suite mates that I lived with for the summer. A little bit of background before I go into my specific project. Environmental enrichment is something that Dr. Craig's lab focuses on as well. It is voluntarily enhanced physical, social, and intellectual activity. Specifically, they also look at exosomes, which are naturally produced, non-inflammatory, biologically active vesicles, and they're able to cross the blood-brain barrier, which makes them important for possible treatment options. You can see exosomes here, released from a donor cell and gone to a recipient cell. The exosomes carry proteins, RNA, and DNA and are believed to influence the recipient cell via these contents. Myelin is the fatty insulation surrounding the neuronal axons. You can see here is an axon with the neuronal or the myelin sheath surrounding it. Oligodendrocytes are cells which wrap the myelin sheath around the axon. Myelin enables faster conduction of nerve impulses within the brain. Multiple sclerosis is when demyelination is intensified in multiple sclerosis patients. Demyelination is common with aging in general, however, in multiple sclerosis is much worse. You can see here demyelination in progress. Demyelination makes it harder to do nerve impulses, and to compound this, multiple sclerosis also decreases the ability to remyelinate. This damage has been shown to occur both in gray and white matter within the brain. My specific project was to see if exposure to environmental enrichment will increase compact myelin in both gray and white matter, and if environmental enrichment serum-derived exosomes will preferentially traffic to oligodendrocytes. Here's what I did. I was able to use confocal microscopy, which is shown here. The exosomes which we isolated were tagged with a red fluorescent tag. Here are the cells we tagged, specifically in this one is microglia, which we tagged green. The confocal microscopy enables you to see 3D images so we can see the exosomes within the cells. Group A is from exosomes from non-enriched rats. Group B is from enriched rats. The first columns here in red show the exosomes where they went. And then we have in the green the cells. Specifically, we looked at oligodendrocytes, microglia, astrocytes, and neurons. The arrows indicate places where exosomes went into cells. And as you can see here, there's one there, a very good one there. For our results, we found that gray matter, which is found here on the outside of the brain, and then the white matter, which is found on the inside. We found that gray matter and white matter from EE brains had increased compact myelin. We used fluorostaining, and you can see here where it's red and white is increased areas of compact myelin, whereas areas of blue indicate low concentrations of compact myelin. Both EE brains, as you can see here, had significantly greater amounts of compact myelin than the NE brains shown here and here. We also want to look at the trafficking patterns of exosomes from NE and EE derived. And we found that in respect to comparison between EE and NE exosomes, there was statistically significant increase in EE exosomes going to oligodendrocytes in comparison to non-enriched exosomes going into oligodendrocytes. We thought this was interesting because oligodendrocytes are involved in myelination, as I had mentioned earlier, and thus this could explain why the environmentally enriched rat brains showed higher amounts of compact myelin. You can see here examples of the cells, the oligodendrocyte wrapping around the axon, the neuron, the microglia, and the astrocyte. The implications of our findings: environmental enrichment increases compact myelin gray matter is important because no one has shown up in the past. It has always been shown that white matter has increase in compact myelin due to EE. Myelin decreases in gray matter in multiple sclerosis, thus EE could be used as a possible treatment for multiple sclerosis. Environmentally enriched exosomes have different trafficking patterns than the non-enriched exosomes, which helps us find parts of the pathway by which EE increases myelin. From this summer, intellectually I learned immunohistochemistry, which was the tagging with the fluorescent tags. I learned about Western blots, and I also learned that if you touch the blots, you get a really pretty picture of your thumb instead of your data. I also learned how to write scientific research papers, as well as how to present my findings in a poster. Here you can see me at the end of the summer with my poster. Professionally and personally, I enhanced my lab skills through my work this summer, and I also got experience working with different machines and tests than I do here at Cornell. I also had the ability to shadow an anesthesiologist and got to witness the birth of twin girls via C-section. Academic impact is I was able to apply the skills I've learned at Cornell in the lab setting, and I also was able to find an opportunity for my honors thesis. The professional impact: I was able to meet and form relationships with medical school admission staff at the University of Chicago. And I also learned that direct patient care is something I definitely want as part of my career. The future impact for me: I plan on returning to Dr. Craig's lab in the future and continuing my research. It also provided me with background behind medicines and treatments and kind of the steps by which we have to be taken to get to the medicine we use today. It also helped me solidify my career choice to become a medical doctor. I would like to thank Becker, the donor for my summer research, as well as my site supervisor, Dr. Richard Craig, my faculty sponsor, Barbara Christie Pope, and everyone else in Dr. Craig's lab.